We're going to explain what monoclonal antibodies are. Before we talk about monoclonal antibodies, let's talk about antibodies more generally. Okay, so I've drawn here, this is a picture of a drawing of an antibody molecule here. Okay, and this is it's a protein. And what it will do is it recognizes, it's got two recognition sites. It's got one here, like a Y, and one on the other side of the Y there. Okay, and they will recognize very specifically a target protein. They recognize that protein and no other proteins. Okay, so for example, if you've got antibodies to the coronavirus, it is probably recognizing, it will bind to there's a protein from the virus. It will recognize the spike protein on the virus, and this one can also recognize the spike protein on the virus. Okay. Right, so if we, let's see what happens here. Then we have cells in our body called lymphocytes, which are white blood cells. And we have millions of different types of these, and each one can recognize a different protein. Some of them may never actually get around to recognizing that protein. They just circulate in your blood, waiting for it to come into contact with it. But if you do come into contact with that protein, like the spike protein, the lymphocyte will be activated. It will divide many times, and it will produce a lot of these cells called plasma cells. And they look different to the lymphocytes and the ones they descended from, and they are just like antibody factories. They produce vast amounts of antibodies, and they churn out the antibodies into the bloodstream and the tissues, and the antibodies then go and neutralize the foreign proteins. That's how antibodies are made. All right, now let's see how this has been this system has been used by scientists to make monoclonal antibodies, and let's see why monoclonal antibodies are so useful. Okay, so what they do is, let's think about a pregnancy test. When females are pregnant, in the first 3 months, they produce a hormone called HCG, human chorionigonadotrophic hormone. Okay, it's a protein that you only produce when a woman is pregnant for the first 3 months. Right, so if you can show that that is in her blood, or very small amounts in her urine, you can show that she must be pregnant. Okay, so that's a protein. So that is actually the HCG is purified, and it's introduced into a mouse. Okay, now the mouse will recognize that protein as being foreign. It will make a load. Its lymphocytes will divide, and you'll end up with a load of plasma cells, which are making antibodies to that protein. Okay, and each one of those plasma cells may be producing a slightly different sort of antibody, but they all recognize the protein. So these plasma cells, they're harvested from the mouse blood, and they're mixed up with some of these red cells I've drawn here. Now these red cells are tumor cells. Now the reason why they use tumor cells is because tumor cells will carry on dividing and dividing and dividing. Whereas if you just get plasma cells from the mouse's blood, they're not cancerous, so they won't divide. They'll just maybe divide once or twice, but then they'll stop. Tumor cells will carry on dividing forever, and you want to produce a cell which will carry on dividing forever. So that's why they get the tumor cells, and they mix these two cells up, and they add a chemical to allow the cell membranes to mix together, and you get these cells. You get this. Some of these cells here are called hybridomas. That just means a mixture of two cell. Hybrid mixture, yeah. And you can see that. So when you mix the two cells together, you get some of the black ones. They're plasma cells that haven't managed to join up with a tumor cell. You get the tumor cells, the red ones, some of them that haven't managed to join up, but then you get a few of them, these so-called hybridomas, where it's a mixture of the two different cell types. And they're really useful because what they can do is they can make antibody, and that property comes from the plasma cell, but they can also divide forever, divide as much as they like. So now you have to try and somehow get rid of all these plasma cells that you don't want and all these non-fused tumor cells. So you just get the hybridomas. Now, how is that achieved? Okay, well, that is quite clever. Well, the plasma cells are quite easy to get rid of because they will just die. They can't divide. So if you just leave them in the tissue culture for a bit, they'll just die. The tumor cells, they won't just die, but these tumor cells have a specific deficiency, and they can't make a chemical that they need to make one of the bases of DNA, as it happens. They need a supplement to the tissue culture medium. Without that supplement, they won't grow. So you have a special tissue culture medium without this supplement. So what happens is all of these will all stop. They'll die. And of course, the hybridomas won't die because they've got the genes from the plasma cells, so they don't need the supplement. Okay, so then you get these cells and they're plated onto a Petri dish with tissue cultures, and what will happen is you'll get little clones of cells growing all over it. And each one of those little plates is derived from one cell that's divided and divided and divided. Now, they're picked off and they are put into a flask here. And if you're lucky, you will pick off a hybridoma. So this is like this is a hybridoma colony, all from one cell. That's making an antibody. That's making the antibody that you're interested in, an antibody which is going to react with the protein we originally injected the mouse with, the HCG protein. Okay, so they're growing in tissue culture. Those cells like to stick to the bottom of the plastic dish, as I've drawn there in red. And so here, that they secrete very large amounts of the antibody. So you've got lots of antibody there in the tissue culture medium, and that is the monoclonal antibody. So of course that is tested to make sure it does react with the protein that you're interested in, the HCG protein. And then those cells can carry on growing and growing, dividing as much as you like. So as long as you don't let them die, you have a continual little factory that can make this antibody anytime you want it. Okay, and that antibody has come from one cell. So that's why it's called a monoclonal antibody. It's come from a clone of cells which have all been derived from one cell, and they're all producing exactly the same molecule, exactly the same antibody molecule. Right, so what can you do with those antibody molecules? Well, let's have a look. Well, first of all, so number one then, you can test for the presence or the absence of a protein, e.g. in a pregnancy test, you're testing for HCG. Okay, so the way that is done is the antibody, so it's like a Y shape. This end is going to bind the HCG here, and that one will also bind it. And what is usually done is on the other end, it's tagged with, should we run that in maybe a brighter color. This is a fluorescent molecule. So in the pregnancy test strip, you get it's dipped in the urine, and there's lots of these antibodies which will bind to the HCG if it's present in the urine, and then those, because they'll stick there, all the other ones that haven't stuck to the HCG will get washed off, and so you'll end up with a colored part of the strip. Okay, so now this can then be expanded a little bit. You can use it to quantify how much of a protein you've got, not only just say it's there or not there, and that's because you've got this colored or fluorescent molecule on the end, so you basically just measure how strong how intense is the color. Okay, and the amount of color you've got is proportional to the amount of protein you've got. So that'll tell you how much you've got there. So you could see if somebody is, you could measure a patient's blood to see if they're diabetic, to see are they producing any insulin, are they producing inadequate amounts of insulin. You measure the amount they're producing. Okay, and finally, monoclonal antibodies can be used in the treatment of diseases. A good example, they're predominantly used in treating cancers. Okay, so we'll talk about one called Herceptin, which is in the news quite a lot because it's very expensive, and that's used for treating breast cancer. Okay, so what Herceptin does, it's a monoclonal antibody that recognizes something on the surface, a protein on the surface only of cancer cells. So it will leave all the other cells alone and it will bind to the cancer cells. Now this it's not entirely clear how that ends up in the cancer cell dying, but it does end up dying, and it's probably because it then recruits the rest of your immune system to go along and kill those cells which have been picked out by the antibody. There are other ways of doing it. Sometimes they don't just give an antibody; they give an antibody to something found on a cancer cell plus a toxic molecule. So you don't want that toxic molecule going around to all of the cells in the body because it will cause toxic effects on them, cause side effects. It will only deliver them to the cells which are actually the ones you want to kill, the cancer cells. Sometimes instead of a toxic molecule, they use a radioactive, something which is radioactive. And again, the radioactivity is too dilute in the rest of the body to cause many problems, but it will concentrate where the cancer cells are and kill them. Another treatment of diseases, there's one which is used for treating Crohn's disease, which you may have heard of. And Crohn's disease is when your own immune system attacks your intestines. And the way this works, you make an antibody to a molecule which stimulates your immune system. So this molecule is called tumor necrosis factor, or TNF. It stimulates your immune system. If you can make antibodies to that, the immune system isn't stimulated, and that stops your own immune system attacking the cells of your intestine. And finally, there's another use for them, where they've been used in research, where you can add them to microscope slides. Yeah, so you add antibodies to the microscope slide. You've got a slice of some tissue, and you, for example, if you had a bit of the pancreas, and you want to see where is insulin made, so you make antibodies to insulin. You put the monoclonal, you put a colored marker onto the monoclonal antibody. You put that onto the microscope slide, and it will color only those cells which contain insulin, because it will only bind to those cells with the insulin. It won't bind to the other ones, so it will show you exactly where in the pancreas where the insulin is being produced.